Welcome to StudentPal. In this Grade 12 Biotechnology lesson, we begin Chapter 9, Stem Cell Culture and Organ Culture. Stem cells are remarkable because they can renew themselves while retaining the capacity to produce specialized descendants. Organ culture and organoids add another level by preserving or rebuilding selected three-dimensional relationships between cells. These systems help us investigate development, disease, drug response, and possible regenerative applications. Yet, attractive images or a few molecular markers do not prove stemness, potency, maturity, or clinical usefulness. Every claim needs functional evidence, appropriate controls, stable cell identity, and a clear statement of limitations. By the end, you will be able to classify potency, compare stem cell sources, explain directed differentiation, evaluate tissue models, and organize a responsible CBSE answer from biological mechanism to evidence and risk. Our roadmap begins with the two defining properties of a stem cell: long-term self-renewal and regulated differentiation. We then build a potency hierarchy from totipotent to pluripotent, multipotent, and unipotent states, always treating potency as a tested capacity rather than a label. We compare embryonic, adult or tissue-specific, perinatal and induced pluripotent stem cells by source, developmental range, practical limitation, and ethical context. Next, we study how substrates, media, signaling, density, and passaging maintain an undifferentiated population, and how time developmental cues direct differentiation. We then compare dispersed cell culture, organ culture, and organoids. Finally, we trace regenerative translation through identity, purity, function, delivery, immune compatibility, tumor risk, long-term monitoring, consent, and evidence-based communication. The central question is an evidence question. A cell that expresses one stem cell marker is not automatically a functional stem cell. Self-renewal requires evidence that the population can generate daughter cells over repeated divisions while retaining the relevant state. Differentiation requires evidence that descendants acquire a specified lineage identity and perform an appropriate function. A three-dimensional structure that resembles an organ is not automatically an organ equivalent. Its cell types, spatial organization, maturity, and measurable function must be characterized. Good experimental reasoning identifies the starting population, treatment, comparison control, direct observation, and justified inference. It also checks contamination, chromosome or genomic stability, and passage history. The conclusion must never extend beyond the resolution, duration, and biological completeness of the model. Stemness is not simply rapid cell division. During self-renewal, a stem cell divides in a way that maintains the stem cell population. Division may be symmetric, producing two stem-like daughters or two committed daughters, or asymmetric, producing one stem-like cell and one cell beginning differentiation. The balance depends on intrinsic gene regulatory networks and signals from the surrounding niche or culture environment. Differentiation changes gene expression, structure, and function as cells progress through progenitor states toward a specialized lineage. Evidence should therefore combine long-term propagation with retained differentiation capacity. Colony formation, lineage tracing, serial transplantation where appropriate, defined marker panels, and functional tests provide different kinds of support. No single marker or short observation establishes both self-renewal and developmental potential. Potency describes the range of cell types a cell can generate under appropriate conditions. Totipotent cells have the broadest capacity. They can contribute to embryonic as well as extra-embryonic tissues. Pluripotent cells can produce derivatives of ectoderm, mesoderm, and endoderm, but they do not independently form an entire organism with its extra-embryonic support. Multipotent stem cells generate a related family of cell types, such as the blood lineages produced from hematopoietic stem cells. Unipotent progenitors mainly generate one lineage while retaining some renewal ability. Potency is not the same as normal function, safety, or clinical value. A potency claim requires validated differentiation evidence, suitable positive and negative controls, and proof that apparent lineage markers correspond to genuine cellular identity and performance. Embryonic stem cells are derived from an early developmental stage and can show pluripotency, but their acquisition raises embryo-related ethical and regulatory questions. Adult or tissue-specific stem cells maintain and repair particular tissues. They may be more restricted and difficult to expand, yet they can be closely connected to normal tissue biology. Perinatal sources such as cord-associated tissues offer additional populations with source-specific properties. Induced pluripotent stem cells or iPS cells are produced by reprogramming differentiated somatic cells so that they regain a pluripotent-like state. They enable patient-specific disease models and avoid some embryo-related issues, but reprogramming does not erase every risk. Genetic or epigenetic abnormalities, residual source memory, clonal variation and incomplete differentiation require testing. The best source depends on the biological question, not potency alone. Stem cell culture is an active control system. The substrate supplies attachment and mechanical information, the medium supplies nutrients and carefully selected signaling factors. Cell density influences cell-to-cell communication, survival and spontaneous differentiation. Colonies or populations are inspected for expected morphology, but appearance is only an early warning signal. Passaging must preserve viability and avoid selecting abnormal clones. Culture conditions designed for self-renewal suppress or balance differentiation pathways, while a differentiation protocol deliberately changes those signals. Quality control includes authenticated identity, sterility and mycoplasma testing, passage history, chromosome or genome stability, marker panels and functional potency assays. These tests answer different questions. A culture can look normal while carrying a chromosome abnormality, or express markers while losing the ability to produce a functional lineage. To test self-renewal, begin with a defined cell population, and where the question requires it, a single cell-derived clone. Record the initial identity and culture conditions. Allow the cells to proliferate, passage them repeatedly, and calculate expansion or population doublings. At later stages, test whether the cells still retain the expected stem cell state and can generate the required lineages. A temporary burst of proliferation is insufficient because committed progenitors may divide several times without long-term renewal. Controls include a known stem cell population, a differentiated or lineage-restricted comparison, and untreated or standard condition cultures. The experiment must also exclude selective survival of an abnormal subclone. Serial propagation combined with retained developmental function supports self-renewal more strongly than marker staining at one time point. Directed differentiation uses knowledge of embryonic development to guide cells toward a desired fate. The protocol does not simply add one final marker-producing chemical. Cells first become competent to respond, then receive signals that specify a broad lineage, commit to a narrower progenitor state, and finally mature toward a functional cell type. Signal identity, concentration, duration, order, cell density, and substrate all influence the outcome. For example, a pathway that promotes one fate early may inhibit maturation if it remains active too long. A strong experimental diagram shows the starting cells, successive signaling stages, intermediate identities, and final functional test. Parallel untreated or alternative lineage controls reveal whether the changes are specific. Batch variation and asynchronous differentiation mean that final cultures often contain mixtures rather than one pure cell type. Differentiation efficiency is the fraction of starting or recovered cells that enter the intended lineage, while purity is the fraction of the final culture belonging to that lineage. Neither measure proves maturity. Identity should use several complementary markers, preferably including the absence of inappropriate or undifferentiated cell markers. Structure and gene expression add evidence, but the decisive test is function appropriate to the target cell, contraction for cardiomyocyte-like cells, regulated secretion for endocrine cells, or electrical properties for neurons, for example. Residual undifferentiated cells are important because they may proliferate uncontrollably after transplantation. Purification or selective enrichment can reduce this risk, followed by retesting. A complete conclusion reports efficiency, purity, maturity, stability, and assay limitations, rather than saying simply that differentiation was successful. Organ culture maintains a tissue or small organ fragment outside the body, while preserving some native architecture, extracellular matrix, and communication between cell types. This can reveal responses that isolated cells cannot reproduce. The tissue may be supported at an air-liquid interface, on a membrane, or within another validated system that allows nutrients and gases to reach it. Diffusion is a central limitation because material must travel from the medium into the tissue without a functioning circulation. Thick regions may become oxygen or nutrient limited, and dissection itself causes injury. Endocrine, immune, and nervous inputs from the whole organism may be absent. Therefore, organ culture is more structurally complete than dispersed cell culture, but still represents a controlled fragment with limited duration and physiological scope. Organoids are three-dimensional cultures in which stem or progenitor cells self-organize into structures that reproduce selected features of an organ. The cells interact with the supporting matrix and a sequence of signals that permits growth and spatial patterning. Intestinal, brain, kidney, and other organoid systems can model development, inherited disease, infection, and drug response. Patient-derived organoids may preserve useful genetic variation. However, an organoid is not a miniature fully equivalent organ. It may lack blood vessels, immune cells, nerves, mechanical forces, or complete maturation. Size, shape, and cell composition can vary between batches. Evaluation therefore requires defined structural criteria, multiple cell type markers, spatial measurements, and functional assays. The claim should name the feature reproduced, rather than treating visual resemblance as complete organ function. Model selection begins with the question. A dispersed cell culture offers excellent access, controlled exposure, and relatively simple measurement, but it loses tissue organization. Organ culture retains native relationships and extracellular matrix for a limited period, although diffusion and tissue injury complicate interpretation. Organoids can be expanded and can reproduce selected developmental or disease features, but their composition, maturation, and architecture may vary. More complex does not automatically mean more valid. The experiment should choose the simplest model that contains the biological feature needed to answer the question. Important findings can then be tested across complementary models. In an exam comparison, organize the answer by starting material, architecture, cell diversity, control, duration, major application, and limitation. That parallel structure earns clarity as well as marks. Regenerative biotechnology aims to replace, repair, or support damaged tissue using cells, engineered tissues, or factors that stimulate repair. Translation begins with a reproducible manufacturing process and a well-characterized starting population. The final product must meet identity, purity, viability, potency, and stability criteria. Delivery must place cells where they can survive and act without moving to unwanted sites. Immune compatibility may require matching, immune management, or other validated strategies. Preclinical studies assess dose, biodistribution, ectopic tissue formation, infection, genetic instability, and tumor risk as well as desired function. Clinical evaluation then asks whether the benefit is real, durable, and greater than the risk. A laboratory demonstration of a marker or short-term improvement is only an early step, not proof of an established treatment. A stem cell-derived transplant can fail in several ways. The recipient may reject the cells or develop harmful inflammation. Contaminating organisms can be transferred with the product. Residual undifferentiated cells or genetically unstable clones may form unwanted growths. Correctly differentiated cells may migrate, lodge, or produce tissue in an inappropriate location. Cells may also die, fail to integrate, or lose function. Risk control begins before transplantation through qualified raw materials, controlled manufacturing, identity and purity assays, genetic stability checks, sterility testing, and a potency assay linked to the intended function. Preclinical studies examine dose, route, and biodistribution. Long-term follow-up matters because some adverse outcomes appear slowly. A risk control matrix should pair each named hazard with a prevention step, detection method, and response plan. Ethical assessment begins with the source of the cells. Donors need appropriate information and consent, and genomic data may require strong privacy protection. Embryonic sources involve additional moral, legal, and regulatory questions that differ between jurisdictions. Ownership, commercial use, and equitable access also matter. For patients, hope must not be converted into misleading certainty. A clinic or advertisement that uses scientific words, testimonials, or before and after images without controlled trials does not establish effectiveness. Students should look for a defined product, approved oversight, transparent evidence, realistic risks, and long-term monitoring. Avoid giving clinical advice from classroom material. Responsible biotechnology combines scientific validity, consent, safety, privacy, fairness, and honest communication. Ethical quality is not an extra paragraph after the science. It shapes the entire path from donation to application. For a CBSE answer, define stem cells using both self-renewal and differentiation. When explaining potency, arrange totipotent, pluripotent, multipotent, and unipotent states from broad to restricted and give the developmental range, not just examples. Compare stem cell sources in parallel columns for source, potency, use, limitation, and ethical issue. Draw directed differentiation as a staged arrow sequence with signals, intermediate progenitors, and the final functional test. For organ culture and organoids, mention preserved architecture but also diffusion, vascular absence, maturity, and missing whole body interactions. In an application answer, trace manufacturing, identity, purity, potency, delivery, immune compatibility, tumor risk, and monitoring. Always distinguish observed markers from inferred function, include an appropriate control, and limit the conclusion to the model tested. Close your notes and rebuild the chapter. Stem cells combine self-renewal with regulated differentiation. Totipotent, pluripotent, multipotent, and unipotent states describe progressively restricted developmental capacity that must be tested. Embryonic, adult, perinatal, and induced pluripotent sources differ in biology, practicality, and ethics. Define substrates, media, signals, density, and passage, maintain cultures, while identity, sterility, genetic stability, and potency require monitoring. Directed differentiation follows developmental timing from competence through specification, commitment, and maturation. Identity, purity, maturity, and function are separate measurements. Organ culture preserves native tissue relationships but is limited by injury, diffusion, and missing circulation. Organoids self-organize and reproduce selected features, not complete organs. Regenerative translation requires controlled manufacturing, delivery, immune compatibility, tumor risk assessment, and long-term evidence. Consent, privacy, fair access, and honest claims complete responsible biotechnology.